Bismillahirrahmanirrahim. Welcome back, friends. Welcome back to another video lecture by Kami Microbiologist. Today in this video lecture I am going to talk about the B cell development and their maturation. In the previous video lecture, if you guys remember, we talked about the B cell receptor and we talked about the B cell biology overview. We also talked about the T cell development and T cell maturation in detail. Okay, so before starting the video lecture about the B cell development and maturation, you need to understand about the B cell receptor, B cell biology and T cell development and their maturation also. If you missed these video lectures, so here I will recommend you that first of all watch this video lecture, due to which your concept will be clear. After that, then this one video will be very easy for you. Okay, so without wasting the time, let's start the video. So we are discussing about the B cell development and their maturation. Okay, first of all, let me give you a little bit idea about the B cell development and their maturation. Then we will explain it with the help of diagram. B cell development is the process in which the B cells are trained or mature, or we can say the B cells are arises, or where the B cells are formed. So this process is called as B cell development. There are several stages, several cells involved for the B cell development, and the B cell development are mainly occur inside the bone marrow. Inside the bone marrow there are different types of the stem cells, and we have different other cells due to which we get the B cells. Now the B cell which we get from the bone marrow, it's immature B cell, and it need to be mature, it need to be trained, it need to be functionable or workable for the disease, or if there is disease condition. At that purpose it need to be mature. So what happened, this B cell actually mature with the help of neutrophils. Neutrophil actually secreted, or they first of all engulf a kinds of microbes and digest it and convert the microbes into small fragments. After that they will release each fragments of the microbes, and once the B cell connect with any one of the fragment of the microbes, so then this B cell will get activated. Not this B cell will get activated, but we can say this B cell will be matured, and they know that what type of antigen are bind to my receptor. After that this B cell will be mature and perform their activity. Okay, but after that we can also say that these B cells will be activated and converted into plasma cell, and after that the B cell will perform their function. So this is a little bit story about the B cell development and their maturation. So now let me explain with the help of diagram. Let me remove. Okay friends, so now let me write. Okay, so B cell development and maturation. As I already told you that B cells are mainly developed inside the bone marrow. So let me draw the bone marrow here. So okay friend, so now let's suppose this is the bone marrow. Okay, inside the bone marrow we have first cell which we call that uncommunicative pluripotent stem cell. So let me draw. So this is the first cell. This cell is called as uncommunicative pluripotent stem cell. This cell will be converted into another cell which we call it commutative pluripotent hematopoietic stem cell. This cell will be converted into this cell will be converted into hematopoietic stem cell. This hematopoietic stem cell will be converted into pluripotent stem cell. And after that, this pluripotent stem cell will be converted into lymphoid stem cell. So this is the lymphoid stem cell. Now this lymphoid stem cell, after that it will be converted into lymphoblast. This is lymphoblast. From this lymphoblast we get the cell. So after that we get another cell which is the immature form of the cell, and we call that prepro B cell. Another cell. So this is called as prepro B cell. Okay. It means that it is the B cell, but they don't have all the receptor. They have a receptor, but in a very less amount. And they have also a genetic material, each and everything, but not in a mature form of the B cell. This is just the B cell, formed, but in immature stage. After that, this pre-pro B cells, what actually it will do, this pre-pro B cell will contact with another cell which is present inside the bone marrow. We call that a stromatal cell. So remember guys, all these processes occur inside the bone marrow. After that, these B cells, pre-pro B cells will contact with stromatal cell. So remember guys, this is the stromatal cell. Let me write it. This stromatal cell have a receptor which we call that VCAM1. So this stromatal cell have a receptor. Let me draw the receptor here. This receptor is called as VCAM1. On the other hand, these B cells have a receptor which we call that BLA4. So this VLA4 mainly bind with this VCAM. So this is what this is, the VLA4. VLA4. Okay. So once these pre-pro B cells and stromatal cell are connected with each other via the receptor which we call that VCAM1 from the stromatal cell and VLA4 from pre-pro B cells. After that, what happened? The changes occur in the chromosomes of the B cell as well as the stromatal cell. When the changes occur inside the chromosomes of the B cell, so then it will produce another receptor which we call that c-KIT. So this is what? This is another receptor which is called as c-KIT. And the chromosomal changes which is occur inside the stromatal cell due to this VLA4 and VCAM1 recognition, then it will produce another receptor, co-receptor molecule which we call it SCF. So let me draw. So this is another, this is called as SCF. So when both recognition occur, so it means that both cells are, strongly recognized each other. And due to this way, there is also some other changes occur inside the B cell. And inside the chromosome of the B cell, there is the heavy chain genes are also present, which is nothing but a kind of receptor genes, or we can say antibody genes are present inside this B cell. These genes are, modified and changed by this recombination, as you can see, by this recognition of the cell. Once this recognition done, then what happen? This stromatal cell will secrete some kind of cytokine, as you can see, that bind to the receptor of the B cell. Okay. These cytokines are called as CXCL12. So let me once again tell you, when the recognition occur, the pre-pro B cell and stromatal cell recognition occur via the c-KIT and SCF. So then what happened? This stromatal cell, there are some changes occur inside the stromatal cell which they will produce a cytokine which we call it CXCL12. These cytokines will bind on the receptor of the pre-pro B cell. These cytokines will cause some changes inside the B cell. Inside the B cell, there is the genes of the heavy chain of the antibody or of the receptor are present. When each kind of changes occur, so then the recombination of these genes are also happen with the help of RAG1 genes. It means that we have some, let's suppose this is the genes of the B cell. Let's suppose inside this DNA there is specific area where the genes are present, and these genes are actually VDJ or heavy chain genes present. When the cytokines are bind to the receptor of the B cell, so then what happen, there will be a recombination of VDJ genes occur with the help of another product which we call a RAG1 product. This RAG1 product, and with the help of this CXCL12, due to this way the genes recombination will occur, and we can say that VDJ recombination will occur, and it will specially responsible for the heavy chain formation. This is how the process is done. After this process, this prepro B cell will get separated from this stromatal cell. This is actually the first stromatal cell. From this first stromatal cell and contact with the next stromatal cell. So now let me draw here another prepro B cells. So let's suppose this is again the So once these B cell are separated from this first stromatal cell, so then this B cell will contact to another stromatal cell. So let me draw the stromatal cell here also. Okay. Now again this prepro B cell will contact with the VCAM of stromatal cell. So this is again what? This is again VCAM, and with the help of VLA4 of the B cell. So once this recognition is done, after that what happened? This is the second stromatal cell. Okay, stromatal cell. With the second stromatal cell, when the prepro be connected, so then what happened? This stromatal cell will then produce interleukin 7. So they will, after this recognition of the second stromatal cell, so then this stromatal cell will secrete interleukin, which we call that interleukin 7. Okay, so this is actually the receptor of interleukin 7 receptor. Okay, now this is the interleukin that are bind to the receptor of this B cell, which we call, this is actually interleukin 7 receptor. This receptor is also called as a tyrosine kinase, or we call that JAK. Okay, remember guys, when these cytokines are binded, so then this JAK will be phosphorylated. Okay, so this JAK will be what? Phosphorylated. Due to this phosphorylation, it will allow the phosphorylation of STAT5. Okay, so STAT5 is also present here. So this is what? This is the STAT5. STAT5. Okay, so when the phosphorylation of the STAT5 occur, this STAT5 will be migrated to the nucleus portion of the B cell. So let's suppose this is the nucleus portion of the B cell. Okay, so when the STAT5 is phosphorylated, so let's suppose the STAT5 is also phosphorylated. Due to this way, it shifted to the nucleus. Inside the nucleus, we have 3 types of the genes present. So let me draw each types of the genes. This is the first types of the genes. This is the second type of the genes. And we have the third type of the gene also. Okay, so inside the B cell we have 3 types of the genes. This STAT5 phosphorylated form of the STAT5 will first bind to this first genes, and it will transcribe and translate this first genes, due to which we get NMYC and CMYC. So we get these types of product. Okay, now these products are specially responsible for B cell proliferation. Okay, So it mainly responsible for B cell proliferation. So means B cells are proliferated due to this product. After that, the state 5 will also bind to this one genes and transcribe and translate it, and they will make a product which we call that EBF1 product. This EBF1 product are specially responsible to make or to bring changes inside the VDJ recombination, or we can say heavy chain recombination of the antibody. Moreover, this EBF1 product are also bind to this third one genes. When it bind to this third one genes and transcribe and translate it, so then they will produce another product which we call it PEX5. Okay, so what we get EBF1 and PEX5. Both this EBF1 and PEX5 will combinedly make what they will combinedly make the VDJ, means they will combinedly make the B cell receptor on the surface, as you can see. So finally we get, so this is the B cell receptor, which is nothing but just like the antibody. Okay, so we get the B cell receptor, as you can see. Now at that stage, moreover, we have also another receptor molecule we also get, we call that CD25. So due to these types of transcription and translation, what we get CD25 and we get B cell receptor, which is nothing but a kind of antibody. Okay, so these cells, after that we get this type of the cell. Now this type of the cell are the immature cell, but we can say the B cells are not formed. So this is how remember guys, this receptor is also in immature form. This receptor is also not in a mature state. So the whole cell we get completely but not mature in state. Okay, so this is actually the immature B cell. Now once we get immature B cell, so then it's shifted to the lymphoid organ, which is the secondary organ, or again is shifted to the bone marrow for the maturation. But most of the time it's shifted to the lymphoid organ, where it will be matured. Okay, and remember guys, this stage is actually called as late pre-B cell stage. Okay, so this stage is actually called as late pre-B cell stage. So this is all about the B cell development. Inshallah, in the next video lecture, I am going to talk about the B cell maturation in detail, because once the B cells are formed, so then it will be mature or trained. Without mature, the B cell will be not workable, functionable inside the bloodstream when there is disease condition occur. Okay, so B cell should be mature. So how it will be mature, we will talk it later. So please stay tuned and keep watching. Thank you for watching this video.